So the question was, give me an example of a prezygotic isolation. I'm going to lock you guys out in 5, 4, 3, 2, 1. Temporal? I don't. Okay, kind of. Yes. Looking for an illustrative. I should have said illustrative example. So a species of wolves not being able to mate due to one being isolated on an island. That would be prezygotic, right before you get a sperm and an egg together. You know. Fertilization fails to occur. Why, though? That's what I was asking for. Why? What would prevent it? Okay, egg. Well, that's part of the situation, right? Behavioral could cause it, yeah, because you would never come together. That makes sense. They were trying to spell water because I know we're smarter than Watt, but it's not even Watt. I can't even say that. Like, what are you? Yes. Okay. When two species inhabit, oh, that's okay. Yeah. Wow. When two species inhabit a different place, it cannot begin to have. I believe you're trying to spell procreation. Wow. All right. Yeah. Right. Yeah. It's like a sig dig. It doesn't matter. Okay. We'll go with it. I'm good with that. So prezygotic, anything before we make a zygote. And if you forgot what a zygote was, that is after fertilization, you have an egg and a sperm coming together, they make a zygote, right? And so if it prevents the sperm and egg coming together, that would be a prezygotic isolation. Postzygotic would be something else, like maybe pollution. But looking at microevolution in the last chapter, in allele frequency, we have to take it one step further, because if microevolution continues to occur, the inevitable outcome is a new species. And that can be controversial. But really, when you look at your fossil record, and if we ignore humans, it's not. I don't think anybody would argue that we have a completely different variety of species today than we did 65 million years ago. Nobody really argues that. So macroevolution must have occurred, and we can just leave us out of the situation temporarily. Well, that's where temporal was coming in, right? Okay. So macroevolution is best observed within the fossil record. Why? Macroevolution takes a very long time for most creatures. Can you give me an example of something that doesn't need a fossil record to show evolution? What would I not need a fossil record for to view evolution? What type of organism? I could give you a hint, but it would be too easy. I'll let you incubate on it for a minute. Final answer in 5, 4, 3, 2, 1. Okay, so viruses, although not alive, do mutate for sure and do evolve. Okay. Plants, plants are pretty complex. They don't really change as much as you'd think, okay? Now, they do change a little bit, and you can genetically engineer them. That's different. That's artificial selection. But when we talk about macroevolution, getting a new plant, they move at about the same rate as humans do, or other bigger species. But what I'm really looking for are things like the virus, bacteria for sure. Let's leave plants out of it. They're too complex. They're multicellular. Yes, microwave E. coli, probably. Okay, so bacteria is kind of what I was looking for, because viruses technically aren't alive. Bacteria are. And I asked you what organism. Now, bacteria can reproduce hundreds of billions of times in a day. In a 24-hour period, they can get hundreds and billions of themselves. So thinking about that from one, if that were humans, that's probably equivalent to like 100 million years, right? And so we can see single-celled organisms evolve in our own lifetime. And that's how I know, or that's how I not know, but that's how I prove with evidence that evolution does occur, and speciation does occur. And so this requires the origin of species that we call the speciation. So speciation is the final result of changes in the gene's pool, allelic, and genotypic frequency. Remember, the only way we actually know that something has evolved is if the allele frequency shifts. That means P and Q become different from one generation to the next. So we know that if I get an increase in the amount of P or an increase in the amount of Q, things are shifting. Will that alone give me a new trait? No. Okay? What is the source of all new alleles? What is the source of all new alleles? Five, four, three, two, one. You're locked out. So mutations, very good. Okay, like the X-Men, yes. Okay. So I'm glad you guys understand that. That is more than likely going to be on the AP exam. The fact that the only way you get a new version of you is through a mutation. And other than that, you're just juggling around cards that already exist, so to speak. Okay, we can make new versions of us for sure. But you don't make a new allele unless a mutation occurs. And that takes a very long time. So every species has its own evolutionary history. We have our own. And this leads into the species concepts that you guys are going to have to be able to identify. And it refers to the different ways in which a species is defined. Now, if I ask a dog breeder what a species is, they're going to tell me it's a Chihuahua, it's a Great Dane, it's a Pug. And I say, and if you ask a biologist like me, I say, well, those are all the same species, right? Because they can all interbreed and have viable offspring. Even though a Great Dane is like as tall as me, and there's a Chihuahua, which is like an ankle biter. Did I tell you what my daughter did to me at the soccer game the other day? Did I tell you this? It cracked. It was terrible. This lady walked by with a little like Chihuahua dog, and my daughter goes, "Dad, look at that rat." She goes, "Dad, that's not a dog, that's a rat," because she hears me say that all the time. And said it so loud that the lady turned around and looked at me like I was the devil. Because she loved her little dog. She just gave me like the evil hairy eyeball. All right, so how new species evolve? Morphological species concept. Morph meaning to change, difference in structure. So this is based on the analysis of diagnostic traits, distinguishing one species from the other. Now this can be distinguished anatomically, so we can look at the bones, we can look at the organs. And the method was used by Linnaeus. Was Linnaeus correct though? No, he had kind of the right idea, but he went about it way the wrong. He said if we have a giraffe, it will develop genes within that single species to change and morph to its environment. We know that's not possible. The environment chooses the giraffes with the longer necks, the ones that were genetically predispositioned to have long necks, not that you actually create genes based on your environment. But most species are described this way. So when I start to describe something, I look at the physical differences or similarities. So I do use morphological species identifiers to help classify something, but if I really truly want to know what something is, I have to get a little deeper. So the evolutionary species concept distinguishes species from one another based on those morphological structural traits. Why do I have to use morphological traits when I look at, let's say, something like evolutionary evidence? Why would I need to look at morphological traits when I'm digging up fossils? Another way to ask that was, what can I not use if I'm looking at fossils? Yeah, go ahead and do that. So not as many responses. This is a little trickier. You have to dig really deep in the earth for this one, right? Nothing is left but dog bones and dirt. Distinguish species from one another to tell the DNA is missing, right? Okay. So I have to use anatomical fossil evidence. I have to use structure. There's no DNA in a fossil, right? And so, what you guys arguing about what it was? Oh, well, it happens. So, depending on how you thought about that, you could have thought about it a couple different ways. But what I really wanted you to think about is when I look at a fossil, I don't get to use genetics. And that in itself can create a lot of misconceptions. Because we know that there's a lot of things out there that are closely related to each other genetically that don't look anything alike. And so we are limited when we study evolution with fossils. I like to study DNA. I like to look at DNA and the differences in your amino acid sequence and use that evidence for evolution, not fossil records. But fossil records are used. I mean, they're good tools. They give you a rough estimate. So critical traits for distinguishing species are called diagnostic traits. Now, the phylogenetic species concept relies on the identification of species based on a common ancestry. Now, a common ancestor for two or more different groups is what it's referring to. So for us, we'd have to look back to early hominids. Let's go to something not so controversial. How about all bees would have a common ancestor, right? All honeybees, we'll say, would have a common honeybee ancestor. They would have branched off. Do you guys remember phylogeny? When we did taxonomy last year, or the year before? We're going to do that a little bit more. Okay. So when we take a look at this, we're saying, okay, that means that this would be the common ancestor of, I believe that is an alligator. I'm not 100% sure. I cannot read that close. But as we move towards a whale species, becoming a mammal that lives out in the ocean. But this would be the flow of a common ancestor based on the evolutionary species concept, using structure to give phylogenetic relationships too. Now, biological species concept states that populations of the same species breed only among themselves. This is my favorite concept because this one makes sense. If you can mix your genes together and make viable offspring that can also have offspring, then I have something that could survive generation after generation. Therefore, I can show that this is indeed a reproducing species. So these do experience reproductive isolation from other such populations, and by isolating different reproducing populations, I can start to see microevolution and eventually macroevolution. Now, very few species are actually tested for reproductive isolation. That means that usually we don't take species from different isolated environments and mix them together to see if they breed and have viable offspring. That's like if I took a frog from Hawaii and a frog from the Philippines and brought them together and saw if they worked or had babies. They could also have babies. That doesn't happen very often. Now, reproductive isolating mechanisms inhibit gene flow between species. And I asked you about prezygotic ones to start the class off, but there's two types. The prezygotic isolating mechanisms prevent mating attempts or make it unlikely that fertilization will be successful. This can be a lot of different things, but there's habitat isolation where the species occupy different habitats. They'd be like me living in Bay City and my wife being in Florida. We're never going to get together. And then temporal isolation. Each reproduces at a different time. Now, we'll talk about frogs in just a second, but frogs sing at different times, right? Okay, and so because of that, they all reproduce at a different time. Or like, what would be another one? How about different types of deer having a slightly different reproductive season, like elk and deer reproducing at a different time? They're never going to come together. And then there's behavioral isolation, where courtship patterns for recognizing mates happen at different times. That could be the frog singing as well, but it could also be like the birds of paradise dancing. It could be in the spring you hear the partridge beat their chest. You ever hear them do that? No? No? You ever hear that noise in the woods that sounds like a drum in the spring? No? That's a partridge. Sometimes they're in a pear tree. That happens around. Sorry. So mechanical isolation. Yeah, that was a bad one. Mechanical isolation is when you're incompatible That means the animal's genitalia or plant floral structures don't fit. Now, I know where your minds are going there, but let's think about a plant, okay? Let's think about a flower. And so you have orchids, and orchids are much like this. Usually an orchid has one pollinator for that plant, and it fits perfectly into that flower structure. And so that mechanical isolation would only allow one bird or one bee or one type of insect to actually pollinate it. Because of that, that mechanical isolation is going to come into play. There is one orchid, it's a snapdragon. Are you familiar with a snapdragon? The bottom opens up, but it only opens up if a certain insect, and I can't remember what the name of it is, lands on it because it's just the perfect weight to where it bends it down, but doesn't like bend it down so much it falls off it. So it's only one type of bug that can actually land on that and balance there and pollinate. If it's too big and tries to land on it, it just falls right down. It can't hold still. And if it's too light, it's not going to push it down to open it up. So if you ever look at a snapdragon, most of them are like that. So I'll show you a picture of one in a little bit. But then there's gamete isolation, where gametes that meet do not fuse and become a zygote. Sometimes they do fuse, right? And then you get a hybrid, but it's sterile. That can happen as well. Okay? Yep. Like a mule, yeah. So this is an example of temporal isolation. Again, at different times, these frogs are singing. I talk about this one in regular biology. So I will tell you something about all of these frogs. They can all do what, if given the chance? They can all interbreed and make the same. They make a frog. It'd be a different frog, but they can all interbreed and have frogs together. They don't because they all sing at different times of the year. Wood frogs, these are like just after your spring peepers. You ever hear like, you hear that out in the swamps early in the spring? That's usually what you first hear. And then it changes just a little bit. That's the leopard frog. The pickerel frog is a little deeper. The green frog is like, rah, rah, and then the bullfrog is like really deep. Ping. And then you hear the ping. You ever hear the ping? You hear, rah, and then you hear ping. They're calling back to each other. You ever hear them do it? No? All right. Go to the swamp and listen. Not now. They're all in the mud right now. All right? We'll take a field trip. We'll pretend we're wildlife for a day. Sorry. I put that on YouTube. Don't play that for Mr. Urich. All right. So reproductive isolating mechanisms. Post-zygotic is the last one, right? After fertilization takes place. So this can prevent hybrid offspring from developing or breeding. We've talked before about hybrid inviability, meaning it makes a hybrid, but the hybrid is sterile. So the hybrid zygote is not viable and dies or doesn't have more children. So that's where you have hybrid sterility, where the hybrid zygote develops into a sterile adult. I think about the saddest fruit in the store. What is it? It's the seedless watermelon. Never going to have babies. Never. And then we eat them. Oh, how sad is that, right? Or a seedless grape. Isn't that awful? No? All right. I know. Imagine seedless corn. They haven't came up with that one yet. So this is, sorry guys. So this is hybrid. This is I'm laughing at my own self in my head. That's not right. Okay, so here we have a donkey and a horse. They get together and they make a mule. Now, it doesn't matter how many mules you get together. There's no such thing as a baby mule from two mules. Okay? They can try, but they're not going to make viable offspring. Okay? The only way you get another mule is a horse and a donkey. Now, two horses can come together and make a horse. Two donkeys can come together and make another donkey. But when you get these two together, all they will make is a mule, and they come together enough to make offspring, but it's not viable offspring because it cannot have children. It's sterile. Okay. Well, they make mules because they're really strong, okay, and they have the size of a horse, and they have the work ethic of a donkey. Donkeys are kind of hard workers. So they make this kind of a short, stocky individual that they don't have to feed as much. Do you want me to tell you the sad history of mules? You guys, you know how we have salt mines in Detroit? Is everybody aware of that? Like hundreds of feet down in the earth, there's salt mines. This is where we get our salt from. They have They have mule graveyards down there. There are some mules that were born down there, like, and they have never seen the light of day. Or, I mean, they're dead now, but they used to use them for mining. So, yeah, a lot of mules never left the mines. They never saw like even the light. And once we should go to the salt mine. That would be fun. That would be really fun. I've never gone. I've only seen videos of it and stuff. But you can go. Yeah, they take tours. Yeah, you go down the long shaft, and yeah, that'd be fun. All right. I'm gonna stop there. I told you I was only gonna do one section today.